Welcome back, you all. Moving ahead to our episode 4 of the Immunology for Everyone podcast by SKA, Professor Dr. Sunil K Arora, our SME, is here today with a deeper dive into the innate immune system. We had a lot of queries come in via email regarding the hows and the whens of the body's responses. I'm sure a lot of these will be answered in this episode, where cells of the innate immunity, along with inflammation, are introduced and talked about. Keep asking, and I'm sure the picture keeps getting clearer and better. Thank you for the response, and we welcome more questions both via email and comments. Stay tuned, do like, and share these podcasts. Your host, SKA Talks.
Thank you very much. Hello, everyone, and welcome back once again to this very important episode of our podcast. When I plan to talk about the cells of innate immunity and inflammation. Before I go on to discuss all these, I'll just introduce two small things which we are going to talk and refer to time and again in all our subsequent, podcasts. One is like CD markers. I will be talking about cells in reference to some CD markers on them. These are, we know that all leukocytes, they display many surface molecules that are crucial for their sensing of different leukocytes and also related to their function. Now, Kohler and Milstein, they revolutionized this immunology when they discovered the procedure for making of monoclonal antibodies, which means that one type of cell will make one type of antibody. And these monoclonal antibodies, they recognize specific molecules, which you may call as antigens. So these monoclonal antibodies, they revolutionized it. And when this technique came, many labs started making monoclonal antibodies, and sometimes more than one type of monoclonal antibodies from a different lab will recognize the same molecule. That created a lot of confusion around, and that led to the discussion among the researchers to have a uniform system of identification of these monoclonal antibodies so that they can refer to the same molecule. That they started having some kind of discussion in the workshops, which were called the human leukocyte differentiation antigen workshops. The first of such workshop was held in early 1980s when they discussed a terminology which is called the CD, or the cluster of differentiation number, which was assigned to a group or a cluster of monoclonal antibodies which will recognize the same molecule. And so the numbers were allotted to these kind of things, and so these CD numbers started coming up. Till now, we have more than 400 CD numbers which have been, given to all these molecules, and we refer to most of the leukocytes based on CD numbers now. And then I would like also to introduce the major histocompatibility complex. This is a very important complex, and you all will, know about it in my podcast, as well as whosoever knows about immunology, that major histocompatibility complex is a large locus on the vertebrate DNA. It contains a set of closely linked, highly polymorphic genes that code for certain molecules which are called MHC molecules. In humans, we call them as human leukocyte antigen, or HLA. And these molecules, they mediate the interaction of various leukocytes with other leukocytes of the body, and they also determine the donor compatibility of the organ transplant. So you most often hear about them when you talk about the organ transplant match, and that is because of the HLA matching only. And these molecules also determine the susceptibility of one individual to to the various diseases, infections, and autoimmune diseases mainly. So these MHC molecules are actually, you know, they are the information is embedded in the genes, which are called the MHC class 1 genes, class 2 genes, and class 3 genes. Mainly the molecules which are expressed by MHC class 1 and 2, they are called the MHC class 1 molecules and MHC class 2 molecules. These are very important, have relevance in the generation, activation of immune response, both innate as well as adaptive immune response. So now let's come to the topic which we plan to discuss today. So, friends, do you know that there are some striking similarities between a septic shock and the critical COVID disease? You know that about 2 to 3% of people, they landed up in ICU with critical disease of respiratory distress syndrome, fall in the plasma oxygen levels, the blood oxygen levels. This is all very much similar to the septic shock. And both these diseases have a very high mortality rate. The main reason is uncontrolled inflammation, which is accompanied by a cytokine storm, which was became a common world when the COVID was at its peak. So generally, it is also seen such kind of condition happens in those people who are either very young or very old, and also in those who have a compromised immunity because of certain underlying diseases like cancer, comorbidities like diabetes, hypertension, then trauma due to injury or burns. So all these people have immune compromised situation, and these can lead to uncontrolled inflammation. And while sepsis is mainly caused because of the bacterial infection in the blood, but it can happen because of viral or fungal infections also. And the COVID is because of the infection of SARS-CoV-2 infection. So major defect in these are because of the dysregulated functioning of the innate immune cells, because they could not control the inflammation, and that leads to the tissue injury and all the death and everything. So it is very important to understand the functioning of various innate immune cells. So whenever we talk about inflammation, the first thing that comes to our mind is the neutrophil, because neutrophils are the hallmarks of uh, inflammation. Uh, and these are the probably the first cells that are recruited uh, because of the signals or stimuli which are released by the pathogens, which we normally call the noxious stimuli. But where do these noxious stimuli come from? They don't come from the neutrophils only or the pathogen, but they come from the macrophages and other cells which are prominent phagocytic cells which are present in all animals, vertebrates as well as invertebrates. So these cells are known to reside in tissues all over our body, and they are more abundant in peripheral organs in various forms and throughout the body, as we have adipose tissue macrophages in adipose tissue, monocytes in bone marrow and blood, Kupffer cells in liver, sinus histiocytes in lymph nodes, and alveolar macrophages in pulmonary alveoli, so on. So there are macrophages all around our body, and these macrophages, they recognize the pathogen or the pathogen associated molecular patterns via a set of receptors which are expressed on their surface or in their intracellularly also. There are many kinds of receptors, such receptors which are called pattern recognition receptors. They are present, they are expressed out on these cells. The more common are the toll-like receptors, which can recognize bacteria and viruses. There are C-type lectin receptors, which are present in the cytoplasm. There are nod-like receptors which recognize bacteria and cell damage, and they are also intracellular. You have Rig-like receptors which recognize mainly the viruses. There are cytosolic DNA sensors which recognize the DNA sensors. Among toll-like receptors, which are more common and more commonly found in many of these innate cells, and mainly on macrophages, there are many toll-like receptors which are expressed. Some of them are expressed on the surface, like toll-like receptor TLR1, 2, 4, 5, and 6. They are expressed on the surface, while TLR3, 7, 8, and 9, they are expressed inside the endosomes. So they are so widely distributed so that they can recognize the microbial components wherever they are present. And depending on their specificity, like TLR1 and 2, they basically recognize the gram-positive bacteria through their bacterial lipopeptides. TLR4 recognize gram-negative bacteria because of the lipopolysaccharides in their cell walls. Similarly, TLR4 recognize the bacterial flagellin, which also forms the one of these kind of PAMPs. And then we have the viral double-stranded DNA, which is seen by TLR3, and single-stranded DNAs are recognized by TLR7 and 9, 7 and 8. While TLR9 is very unique because it recognizes a sequence of nucleotides which is more frequently found in the prokaryotic DNA. This is called the CPG motif, where we have the flanking purines and pyrimidine nucleotides, and they form the CPG motif. They are found almost 20 times higher in the prokaryotic DNA as compared to the eukaryotic DNA. So they activate the cells through TLR9. So once the PAMPs they bind to the TLRs, it leads to the recruitment of adapter proteins in the intracellular domain of these TLRs, which leads to the activation of transcription factors like NF-kappa B, which translocates into the nucleus, goes to the bind to its binding site on the DNA and leads to the transcription of the following gene, which leads to production of lot of cytokines. And these are the cytokines which are inflammatory cytokines made by the released by the macrophages. And then we have other cells like dendritic cells, which have interferon regulatory factors, and these go and stimulate the transcription of type 1 interferons. So we have acute inflammatory cytokines and the antiviral proteins which are released. And these all pro-inflammatory cytokines, they include IL-1 beta, IL-6, TNF-alpha, type 1 interferons. And these cells, they metabolize arginine to nitric oxide. And along with all these cytokines, they also release many kinds of chemokines. And these are the cytokines which chemo attract other cells to the site of infection or injury. And all these cells like neutrophils, eosinophils, monocytes, many lymphocytes, they all get, and dendritic cells, they all get attracted to the site of inflammation and to the site of infection. And they all lead to a generation of a phenomena which is called inflammation. So these cytokines, they act on endothelial and epithelial cells and leads to release of certain vasoactive amines like histamine, serotonin, prostaglandin E2, leukotriene B4. These all are vasodilators. They leads to vasodilation, which means the capillaries become dilated, and that leads to increase in the permeability. So there is vasodilation, which leads to increase in blood flow, causing redness and warmth. You will see that any area where there is a infection or you have injury, it becomes a little red, swollen, and warm. You can touch it. That is typical of start of inflammation. There is increase in permeability because of dilation that leads to extravasation of exudation of protein rich fluid into the extracellular vascular extravascular spaces, which cause the swelling. So there is fluid exudation, and also there is extravasation of leukocytes, which start rolling, adhering because of the upregulation in the adherent molecules, and they migrate because of increased permeability into the extra vascular spaces. And all these cells, they basically accumulate at the site of inflammation. And so that causes the inflammation. So inflammation is a part of biological response of the body tissues to harmful stimuli, which are the pathogens or pathogen associated molecular patterns, as well as damaged cells, because there is a lot of cells which get damaged because of injury. They also release certain damage associated molecular patterns and some irritants. They all lead to creation of this kind of inflammation. The sequence of events are, say, vasodilation and all these things. So there are five cardinal signs of uh, this inflammation, which include the heat as the area becomes a little warm, pain. It is because it leads to redness of the area, swelling of the area, and sometimes also the loss of function. So these five cardinal signs are very typical of start of inflammation or the inflammation at the site of infection or injury, and this invariably is accompanied by fever. The fever is because of certain inflammatory cytokines like IL-1 beta, IL-6, and TNF alpha. It goes and dysregulates the temperature regulator in our brain and cause this increase in fever. So inflammation is a generic response, and it is considered like a mechanism of innate immunity. The function of inflammation is to eliminate the initial cause of cell injury or clear the damaged cells and tissues, and also initiate the tissue repair. The, if the inflammation is too little, then it could lead to progressive tissue destruction by harmful stimulus, which can be bacteria, and also it can compromise on the survival of these microorganisms, which lead to infection. So we can have inflammation which is called acute inflammation, which is the initial response of the body to harmful stimuli, and this is achieved by the increased movement of plasma and leukocytes from the blood to the injured tissue, and a series of biochemical events that propagates and matures the inflammatory response involving the vascular system and immune system and various cells in the injured tissue. So all these include the acute inflammation, which is basically a response of the body to the infection or injury. Whereas if it is prolonged inflammation, which goes beyond some time, then it is called chronic inflammation, and it happens in many kind of diseases. It leads to progressive shift of the type of cells which are present at the site of inflammation, because a lot of mononuclear cells, they can come over and take part into that chronic inflammation, and many lymphocytes, the T cells, they also come there and they cause that. So we'll talk about chronic inflammation in relation to some diseases which we may discuss when we take up disease-specific immune responses. And these chronic inflammation involves simultaneous destruction and healing of the tissue also. So we have acute inflammation, which is very important, and chronic inflammation. But everything is not good about inflammation. There are some examples of inflammatory diseases which may find, like in the acute inflammation, we see in allergic reactions, peptic ulcer, bacterial pneumonia, sepsis, and critical COVID also, that is a fulminant acute infection. Um, and then chronic inflammation you see in hay fever and some periodontal diseases, atherosclerosis, uh, osteoarthritis. All these are the conditions which happen because of the chronic condition. So, as I said, that neutrophils are the first cell which get recruited, and they are basically the cells which are present in the circulation. You can find about 50 to 70% of the total leukocytes are neutrophils or polymorphonuclear lymphocytes. They get recruited because of all that process we have discussed. And once the tissue uh, they are recruited at the site, they drive the inflammation, and they try to clear the pathogens because they also express all these pattern recognition receptors, and they recognize the PAMPs and DAMPs, and they undergo degranulation because they are the large granular lymphocytes. They carry granules which have the molecules like perforin and granzyme, and as they degranulate, they release those perforin and granzymes, which basically cause the death of the cells. And there are many lethal mechanisms, effector mechanisms in macrophages as well as neutrophils, and all other phagocytic cells like dendritic cells also. They all upregulate the formation of reactive oxygen species, reactive intermediate oxygen intermediates, nitric oxide, and all. So this leads to a temporary increase in the oxygen consumption by these cells, which is called the oxidative burst or the respiratory burst, which leads to formation of all these, lethal oxygen and nitrogen intermediates, which leads to the death of the pathogen, and also removal of all those cells which are abnormal. The neutrophils, over and above, they also undergo a programmed death, which is called by the process of netosis, because there is a programming, and neutrophil chromatin get condensation. It's a evolutionarily conserved cell death, which process is distinctly separate from the apoptosis, which is the normal programmed cell death found in all our body cells. But this netosis includes the modified chromatin, which is bound with all these, reactive oxygen species, which are and all those, molecules which are lethal to the pathogens. So these release of these modified chromatin decorated with all these molecules, it also sends the pathogens around, which are trapped, and these are all taken up by the macrophages and cleared off. So phagocytosis leads to activation of the effector mechanisms as well as release of toxic compounds because of the assembly of NADPH oxidase complex and making of all these intermediates. So once all this process of killing is going on, because of production of all these molecules, the macrophages generally survive. They finish the job and go on to look for some more damaged cells and some more pathogens. Whereas neutrophils, they normally die. So they are short-lived cells. They get activated, they perform their function, and die also. And that's why the pus which is generated at the site of infection or injury normally contains these dead neutrophils, and you can find a little bit of greenish or yellowish tinge in the pus. This is because of an enzyme which is called myeloperoxidase, which is a copper containing enzyme, which is present abundantly in these neutrophils. So those dead neutrophils, they give that color to the pus also. So there is all these things, they happen in the neutrophils, make them a very good, cell of the innate immune response. Then comes the dendritic cells. So as I said, the dendritic cells also come there. They are the cells which are relatively recently described in 1973 by US scientist Ralph Steinman when he was working in Rockefeller University in New York. He found that most non-lymphoid organs and in epithelial surfaces, there are some kind of cells which have long dendrites, and they are not like macrophages. They have a different morphology under the microscope. These dendritic cells, they are also present in all peripheral organs, and they carry all these unique receptors, which make them capable to capture the pathogens and all those pathogen associated molecular patterns. So these cells, which are dendritic cells, which are present in the peripheral organs and the mucous membranes and all, they are called the immature dendritic cells because they do express MHC molecules on their surface, but they do not express many molecules which are required for subsequent, start of the adaptive immune response. But these cells, they have a unique property of migrating from the periphery to the lymph nodes, carrying the antigen along with them. And in between, they get activated and become mature dendritic cells. And when they start expressing very high amount of MHC molecules, they also express many molecules which are called co-stimulatory molecules. And these co-stimulatory molecules are CD83, 80, 86, CD40, which all help them to present the antigen to the T cells in the lymph nodes. So we will talk about that in the next episode. But it is very important to know that these dendritic cells, they basically are a very important connecting link between the innate and the adaptive immune response. So there are different kinds of these dendritic cells. There are some conventional and myeloid dendritic cells. They are performing similar functions. Then in the inflammation, you have some monocyte-derived and isolate monocytes and culture them in the presence of IL-4 and GM-CSF, they transform into dendritic cells. They are called monocyte-derived dendritic cells. But we do have another kind of dendritic cells which are called plasmacytoid dendritic cells. These plasmacytoid dendritic cells have a plasma-like morphology initially, but once they recognize the viral nucleic acids through TLR receptors, which they express are the TLR7 and 9. So once they do that, they have activation of interferon regulatory factors, which lead to expression of type 1 interferons. These plasmacytoid dendritic cells, although they are present in like 0.1 to 0.5% of total leukocytes, but they produce almost 99% of type 1 interferons which are produced. And all these type 1 interferons, they have antiviral properties. And as a innate immune response, they act like antiviral because of type 1 interferons, but they do express many pro-inflammatory cytokines like TNF alpha, and also lead to now it is shown that they can also cross prime CD8 positive cells and CD4 positive cells. So they also form a sort of important link between innate and adaptive immune cells. Then we have NK cells, which are the natural killer cells. They are large granular lymphocytes. They also carry all this lethal machinery of perforin granzymes, and but they kill only those cells which have a missing self, which do not express MHC class 1. And so they have, and They kill most of the tumor cells which downregulate expression of MHC1, and also some virus infected cells, because virus also downmodulate the expression of MHC class 1. We have NK cells, which are all NK cells, they express CD16, but some of the NK cells, one population, they express very high amount of CD50, 56 also. These are the cells which make lot of interferon gamma, whereas those cells which do not express very high level, amount of CD56, they are the main killer cells through other mechanisms, because they express CD16 is a FC gamma receptor and binds to IgG through FC receptor, and IgG binds to the pathogen or the cell, cancer cell, and it leads to antibody dependent cellular cytotoxicity, which is the hallmark of these NK cells. Then we have certain innate lymphoid cells, which are the tissue resident lymphoid cells. They look like lymphocytes, but they do not express any antigen specific receptors, and they crosstalk with the resident tissue cells via cytokines in that microenvironment, and they secrete cytokines that regulate the innate immunity. There are three types of such cells have been recognized. The ILC1s, they also include the NK cells in them, and some ILC1s. They all depend on transcription factor, which is TBET, and they secrete a lot of interferon gamma in response to IL-12 and IL-18, which is secreted by macrophages and dendritic cells, and these help in the inflammation, generation of inflammation also. And then ILC2 cells, they depend on a different transcription factor, which is called the GATA3, and it includes type 2 kind of cytokines like IL-4, 5, IL-9, IL-13 in response to IL-25 and 33. And these cells, they stimulate eosinophils and mediate inflammation. And then we have ILC3 type of cells, which depend on still another transcription factor, which is called ROR gamma T. These cells, they produce a lot of IL-17 and IL-25, as well as GM-CSF, which is granulocyte monocyte colony stimulating factor, and they produce these cytokines in response to type 1, these IL-1 alpha and IL-1 beta and IL-23, and they cause the inflammation also. So all these innate lymphoid cells, they promote inflammation, and they are present in the tissue, and they behave accordingly as per the cytokine milieu. And there is a lot of plasticity seen that one kind of ILC transdifferentiate into another kind depending on the cytokine milieu, and they behave. So once the inflammation is generated, that needs to be resolved. If it is not resolved, it leads to the tissue injury, and then it can cause death also. That's what happens in sepsis, and that's what happens in a critical condition of COVID. So there are macrophages, we call them M2 macrophages. They perform this function very nicely because they release many anti-inflammatory cytokines like IL-10 and TGF-beta, and they cause the resolution of inflammation as well as they help in repair of the damaged tissue. And it has been seen very recently, more recently, that even neutrophils, monocytes, and macrophages, they all basically help in the resolution of inflammation because they store large amounts of glucocorticoid inducible pro-resolving protein, which is called annexin A1. So neutrophils, they also have this protein, the annexin A1, which is stored in the tertiary granules, and they start releasing it once the inflammation needs to be resolved. So these recent studies have also shown that the presence of neutrophils, they govern the active process of resolution to clear the tissue of infiltrated leukocytes and restoring the architecture and function, and thereby driving and enabling the tissue repair, as well as whenever it is appropriate, the tissue regeneration also. So the macrophages, monocytes, neutrophils, they all help in resolution of the tissue, and any dysregulation leads to the damage of the tissue. So I will close this podcast here, and will encourage you guys to read some of the these book by Abbas and some of the references which are shown here, and they will give you more clear ideas of whatever the function of these cells and inflammation also. So till next episode, when we try to cover the antigen presentation and activation of adaptive immune response. So thank you very much, and bye-bye.
Thank you, Professor. So much happens in just the first line of defense still. It's amazing. We will take one question, and we have very little time, if you will. It was shared with us via email. If the innate
Sure.
immune response is so robust, then how do pathogens survive and are still able to cause the infection?
It's a very nice question, and very important one, and very genuine. So everybody should ask this question. We see that many pathogens have developed strategies that to evade this response. And one of such mechanisms is that some pathogens, they avoid being ingested by the phagocytes. So they block the phagocytosis. For example, some gram-positive bacteria, they carry a very thick and slimy polysaccharide coat, which we call as capsule. So that is not recognized by the complement or the phagocytic receptors. So they are not ingested. While some other pathogens, they are phagocytosed, but they avoid being killed. Like you have the example of Mycobacterium tuberculosis or Francisella species or Brucella species. These microorganisms, they prevent the formation of a mature phagosome. Like I told you that the bacteria or the pathogens, they are engulfed in the phagosome, which goes and fuse with the lysosome and forms a phagolysosome, mature phagolysosome. And the lysosome containing all the enzymes and acidic atmosphere, so they kill the bacteria. But if these bacteria, they block the fusion of phagosomes and lysosomes, and they survive. And there are some pathogens which secrete some enzymes also that detoxify the products of respiratory burst. For such pathogens, the first line of defense, like innate immune responses, are not sufficient to clear the infection. And that's why subsequent adaptive immune responses are very much required. So that's how they evade the immune response.
Thank you. Thank you very much. See you soon, listeners.